Buenos días. Vamos a ver qué tanto está, cómo está tu inglés.
Aquí retando.
Después les hago un examen. I'll make your presentation in Spanish, dear Robert, and we'll start, okay? Buenos días para todos. Hoy tenemos el gran placer de tener al doctor Robert Charters como invitado en nuestro seminario extra del día de hoy. El doctor Robert recibió un doctorado en la Universidad Estatal de Washington en genética y biología celular y ha sido un científico investigador del Departamento de Agricultura de los Estados Unidos, la parte de investigación durante 36 años. Su carrera, su investigación se ha centrado en la aplicación de enfoques moleculares, celulares y biotecnológicos a problemas reales de producción de cultivos agrícolas. Su trabajo actual incluye el desarrollo de soluciones de base biológica para problemas emergentes de plagas y patógenos en cultivos, y hacer esto mediante el desarrollo de técnicas novedosas que permitan una rápida implementación de estas soluciones. El doctor Charters ha publicado más de 120 artículos y posee 4 patentes sobre su investigación, 2 de las cuales están bajo licencia. Además, recibió el premio al científico senior del año 2022 del Departamento de Agricultura de Estados Unidos. Actualmente lidera el gran desafío de cítricos del Departamento de Agricultura y codirige con el doctor Michel Heck, también del Departamento de Agricultura en Ítaca, un equipo de científicos del USDA, la universidad y el sector privado, con una financiación que tienen de una parte particular de financiación del USDA que se llama NIFA. Esta financiación se centra en el desarrollo de soluciones para enfermedades de los cítricos, una enfermedad que ha reducido la producción de cítricos en Florida de más del 80% y amenaza a California y Texas. Recientemente el doctor Robert ha regresado a su puesto como líder de investigación para la Unidad de Investigación de Insectos Subtropicales y Horticultura en Fort Pierce, en Florida. Antes de esto, hasta el año pasado, él tuvo un puesto como director del Laboratorio Europeo de Control Biológico del Departamento de Agricultura de los Estados Unidos en Montpellier, en Francia. Entonces, el doctor Charters nos va a presentar el seminario sobre simbiontes y es muy, muy importante para nosotros en Semicafé y para toda la agricultura en Colombia, porque estamos tratando de desarrollar tecnologías para el control de plagas y enfermedades que sean muy innovadoras y que no tengan que ver con el uso necesariamente de químicos, que son, pues, digamos, una de las formas principales en que hoy en día controlamos estas plagas y enfermedades. Entonces, es muy importante esta presentación del doctor Charters. Dr. Robert, the audience is yours. Thank you very much. We really appreciate your time to be here with us today in the likely collaboration we're going to have. We are really excited to hear your talk today. Thank you very much, Dr. Robert.
Thank you, Marco. First of all, you can hear me okay? And
yeah.
And you see the presentation, no problem.
Yeah, it's fine. It's perfect.
Okay. Well, and then second, I would like to thank you and Lily Cano. Lily had put us together in contact, and we started talking and realized that this would be a great way to start out our discussions about potential collaborations on coffee. And I really appreciate the opportunity. So with that, I'll get going. We have been facing a problem in citrus that I'll discuss a little further. That is a pathogen that has come in and really decimated the industry. And it caused us to rethink a lot of things. That plus, as I heard was described in my introduction, you heard I spent 2 years in France as the director of the EBCL, European Biological Control Lab. It's an ARS lab there. And I realized the need for developing biotechnology platforms to deal with invasive pests that keep coming in. We need to do a better job at delivering solutions, and that's what this is about. So it's without what we're trying to do is really develop team structures in what we call a transdisciplinary approach to solve agricultural problems. Okay, found out. I gotta figure out how to get this going here. Excuse me a minute. There we go. Okay. All right. So I've been with ARS for about 37 years, and I have this strong passion for understanding the importance and wanting to be a part of developing closer ties and team structure for developing solutions. And the buzzword for that nowadays is called transdisciplinary approach. And so I get to hop on my little soapbox platform and talk a little bit about my passion of developing strong collaborations. And when we say transdisciplinary, you probably heard the word multidisciplinary, interdisciplinary, and they're just different ways of interaction. But the way where interactions are really close, where we overlap expertise, we work very closely with multiple disciplines in a holistic fashion. That's called transdisciplinary. And that's what I think is really necessary to drive things forward and create new innovative solutions. And I'd like to, before I get into what we're doing today, talk a little higher level and say, there was something that came out because I'm very interested in developing these platform technologies that can be adapted to different cropping systems. And the need for it is kind of spelled out here in this report that came out in 2023 by IPBES. It's a subgroup within the United Nations that developed this global view on the concerns of alien species and their impact on our environment and on our agriculture, and why we can't ignore it and why we need to address it. And this is a multinational report that came in, and as a result of interacting with them, I actually sponsored what was called the International Plant Biosecurity Alliance workshop in Montpellier, France, a year ago, about a year and a half ago. We had people from 8 different countries: Australia, China, England, France, Greece, Italy, Spain, and the US. And we worked together to kind of come up with a strategy, a way of interacting closer to address some of these problems. I think it was a very successful workshop. And the idea was, when we were there, was to develop this concept of this biosecurity toolbox that we all work to develop solutions for. So the meeting was really a team building exercise on that project. And really the focus initially is on insect transmitted plant vascular bacterial diseases and what kind of platforms we could use or advance through this transdisciplinary approach. And really we were focusing at that point on 3 different pathosystems: Xylella as a plant pathogen transmitted by leafhoppers, Phytoplasmas, also pathogens transmitted by leafhoppers, and Candidatus liberibacter transmitted by psyllids. And they affect a number of crops: olives, grapes, citrus, a broad array of crops. And so that's really where I think we need to come in and say, how can we develop common platforms to multiple cropping systems? And which is why I'm working on citrus, but I'm talking to you here, people working on coffee, because I'm excited about the opportunity to create team strategies to see if these technologies we're developing, these platforms we're developing, can be adapted to different cropping systems. So in our workshop, really the idea was to create this toolbox of biosecurity platforms that and address major agricultural challenges that can be for which solutions can come out of. Ones that represent major problems in multiple countries. And so use tools, again, that we can adapt. And that's that's what we did. We came together and really it was a very successful workshop, and some collaborations are still moving forward as a result of this. Okay, so then I'll get off my soapbox on this concept I have of biosecurity platforms and talk about a very specific problem that is close to my home and close to my heart, and that is the problem of citrus greening disease in Florida. When people think of Florida, they think of usually Mickey Mouse and citrus, because in Florida, that's are the big economic drivers. However, citrus doesn't look like this anymore. It looks more like this. And that's because of a disease called citrus greening disease. It's a phloem limit bacteria transmitted by the Asian citrus psyllid. The psyllid came in about 1998, and we started seeing the symptoms of the disease of the citrus greening, which is a bacteria was being vectored by the psyllid about 2005. And if you look at citrus production, the gray line is Florida, and you see bumps where there were freezes, but recovery and hurricanes. And this is a bit of an old slide. I apologize, I haven't updated it, but HLB came in around 2004, and it's been going down, and it's still going down. We are now, commercial production in Florida is down about 90% from what it was. So it's really impacted us. The disease is present in Texas and California. It hasn't advanced heavily into their production areas, but they're very concerned that it will. So from a transdisciplinary approach, we put together a team, really a diverse team of researchers with expertise in all areas, plant pathology, entomology, engineering, horticulture, genetic engineering, and private companies, universities, and USDA and government labs as well, and to develop solutions. Some of the ideas when we said, listen, we have to approach this from a new way, we also wanted to say, listen, we need to get away from the use of broad spectrum pesticides. That's always an underlying goal of the USDA ARS. And this just shows the use of pesticides throughout the world in different countries or regions. And this is, we wanted to avoid making this even a worse problem. So let's look at, when we were saying, what could we do? What kind of new technologies? How could we innovate in this area? We said, okay, it's got to be sustainable, cost effective. It's got to be able to use biological strategies, then not those chemical pesticides, have broad customer acceptance, have streamlined regulatory approval, and be deliverable across a broad spectrum of crops. So that's this platform technology. All right, then also we wanted to be able to deliver it in a shorter period of time. We're looking at, we would like a solution that once it's accepted by the community, we could deliver solutions at least within 5 years of starting the research and adapting technologies. I work in GMO areas. I worked in that most of my life. And from starting on basic research to delivery, especially in a crop like citrus, it took 30 years, and we got to do better. So our goal is something more like 5. So with that said, then I would like to introduce the organism that we've kind of settled on and advanced as the foundation of our strategy, and that is Agrobacterium that causes crown gall disease. You see it on a number of plants. Here I am in France with a sycamore, a Patanus tree with a Agrobacterium-induced gall quite large on this tree, and the plant is doing just fine. That tree's been there, that gall's been there for many years. Here's one in Italy I found on a vine, one in Illinois on a deciduous tree. And this is what we're doing. We're saying, wait a minute, these are called, it's called a pathogen. Agrobacterium causes crown gall disease. But can we rethink how we use it? And we're not the first to rethink how we use it, but we want to use it in a way where we say, hey, these galls we could potentially use as a delivery device for us. Almost like, think of it like a someone with diabetes who has a insulin delivery pump attached to them. We're looking at galls as kind of like a biological version of a delivery device. And I'll explain what I mean by that. So Agrobacterium, first to understand, make sure we're all on the same page. I'm sure most of you know this already, but just to bring it together, it actually is a natural genetic engineer. It's been doing it for millions of years. But we're asking, can we rethink old ideas? So rethink how we use Agrobacterium. Agrobacterium naturally causes disease by entering wound sites on a plant, inserting a very specific piece of DNA called this tDNA, the blue, within this larger TI plasmid. That tDNA is excised when it comes next to a plant cell. It's incorporated in the chromosome of the plant cell, and that causes the formation of a plant wall. And that wall is a house that the bacteria lives in. Part of this tDNA codes for the biosynthesis of plant growth regulators and causes the plant cell to divide and form this gall, but also for the form of produced food for the bacteria to live, survive. This was first kind of worked out back in the late 70s, early 80s. Mary Del Chilton picture here was really the driver of this when they discovered that Agrobacterium was actually delivering DNA to the plant. And so that's how many of us now use it to produce transgenic plants using the strategies they worked out. Basically, anything on that big, large TI plasmid between what they call two borders, the right border and left border, anything between those is inserted into the plant DNA. And normally it produced those auxins and cytokinins and protein with food for the bacteria. But they removed all those and created this binary vector where you can put in anything you want and create transgenic plants. But the team, Michelle Heck, myself, and Marco Patino, a postdoc in my lab at the time, Michelle was an ARS scientist in Ithaca, New York, said, well, wait a minute, what if we just put those auxin and cytokinin biosynthesis genes back in, and we form the gall. Instead of using it to create a whole transgenic plant, we just created transgenic gall. That's it. The rest of the plant is not engineered. And can we get it to do something? So how would we do this? Well, our initial vision was to say, okay, we could inoculate the plant with Agrobacterium. It would do its thing for us. It would deliver DNA to a plant cell. It would incorporate into the genome. And that tDNA it would deliver would produce things we would want it to produce. It would grow into a gall. We would culture that gall, selecting for those cells that are engineered so that they're all producing the molecules we want. And then we would transplant those back onto the crop to form functional, what we were calling biofactories. So these bumps on the side of a tree, and ultimately they would grow with the plant and deliver desired molecules to them. Now, that's where we thought we would go, and that ultimately there may be a use for this, but what happened along the way is we realized we could go straight from delivering the bacteria and sidestep these protocols. And why? Because we could do this. It was shown back in the 1930s that you could cut off these symbiont or these galls and reapply the cells to the plant, and they would grow into a gall. And we can do that in some plants like tomatoes and that we were doing, but we've been having difficulty getting that to be reproducible in citrus. I still think we can, and there are some research going on in that way. But in the meantime, we realized, well, maybe we should just deliver the bacteria. And so that's what I'll be talking about today, where we deliver the bacteria, we form a gall. This is one on citrus with a wild type bacteria in our field. And now the idea is that this gall will produce molecules. We call it a symbiont, by the way, because we want to just distinguish it from a gall that's not necessarily a benefit to the plant. So these are engineered to be a benefit to the plant by producing things that could kill the bacteria. It's the phloem-limited bacteria that causes citrus greening. And these molecules, these green dots represent the proteins, antimicrobial proteins, for example, that could be produced. And so that they would move systemically in the plant, in the vascular system, and kill the bacteria. All right. Why did we come up with this strategy? Well, number one, we don't engineer the whole plant. We engineer this gall. And then it's just the product that moves systemically in the plant. Now, so of course, then the technology is whatever's produced moves into the plant, and we'll have to monitor for the residue, but you have to do that with anything. You could have a gall that would have an extended lifespan, so you don't have to keep reapplying. it could be there for many years potentially. And the nice thing is, from a regulatory perspective, in a GMO, the whole plant is engineered. In ours, just the gall is engineered. Another thing is, these galls, if they fall off or are grabbed by a bird and taken away and deposited somewhere, they die. They can't survive off of their host. They don't form pollen. They don't form fruit. They don't form vegetative propagules that are engineered. So we don't have an escape of any of the modified cells into the environment, and the whole tree is engineered. Again, we're getting away from broad spectrum use of chemical pesticides then, and it could potentially be a one-time application for multiple years. All right, so when we started this journey back about 2018, we realized there were a number of aspects we needed to do. We needed to identify the basically bullets for this gun, if we call this a gun, the defense genes that would work to either induce natural defenses or kill the pathogen. We needed to make sure that the structure and development was proper for that symbiont to develop. Export is something I'll talk about in more detail. When they make something, how do we get efficient export? And some biofactory mass production and delivery. How do we deliver this commercially? Field evaluation and regulatory. Those are all aspects that we had to all put together, and that's why we needed this transdisciplinary team in order to get to this commercialized thing. And then the idea then is to get the gall to grow on the plant. This is tomato, an example of over time when we've inoculated. And so, of course, that's everybody knows they form galls, but now we call them symbionts. And we were at our initial research allowed us to actually win a $15 million 5-year NEFA grant to fund this, to try to move this project forward. And we're in our final year of that. I was initially the project director, but I took a 2-year leave of absence to go to France to direct the European Biological Control Lab there and in Greece. So Michelle Heck stepped up, and she's finishing out as the project director. But we really had expertise in many areas: molecular biology, entomology, and engineering to help us develop delivery strategies, transgenic, genomic editing folks, commercial regulatory expertise, field trial evaluation and outreach in education, and molecule discovery work. So, there we go. I would like to say, how do we do this then? If we're going to add genes, how are we going to do this? What's the method that we use to get agro to deliver? We're lucky, as part of our team was Dr. James Thompson from ARS out in Albany, California. He has developed and published on a system called the gantry system using recombinase technologies. They have a way of adding multiple genes directly into the TI or RI plasmid in this case, but we do it with the TI plasmid as well. And that's that very large megaplasmid in Agrobacterium that has many factors on it, but it also has that tDNA region that's moved into the plant cells. And using the recombinase technology, they built an iterative strategy for delivering multiple genes to the tDNA right on that plasmid. And then so we can engineer different bacteria. And that's important because when we set this up for any plant, we have to optimize the genotype by genotype interaction. That's the genotype of the host and the genotype of the bacteria. We screen a library of agro for ones that form the best quality, initially galls on the plant. And those are the ones we engineer. And using this strategy, we now just directly engineer the TI or RI plasmid. Using this strategy, I'll go to the next slide here, a little more in detail. In the iterative strategy, he uses recombinase technology to integrate one gene at a time, and you can keep adding multiple genes until you get how many ever you want. If you're going to engineer a pathway, you can add multiple. They've delivered up to 70 kilobases of DNA using this strategy because the TI or RI plasmids can tolerate large amounts of DNA, and the tDNA will transfer that into the plant fairly effectively up to about 7 kilobases right now. So that if you think about that and you think, okay, let's say an average gene would be 2 kilobases, which is pretty good size, you can manipulate a large number of genes or a large or very complex metabolic pathways theoretically using this strategy. So James is part of our team. He does a lot of our constructs for us, and that's what we add to the plant. So we can form galls on plants, or symbionts, I should say, and evaluate them. They're very nice as a screening tool. Here's one. This one's producing a ruby colored reporter molecule. We had 3 genes to synthesize the butylene molecule that is the red color in beets. And so we can see if cells are engineered to produce that. But we can also then, it's really turned out to be a nice screening strategy where we can evaluate, in this case, we evaluated a number of potential antimicrobial peptides, AMPs, and we've identified some where this is the relative detection based on PCR of the pathogen in plants containing that we produced these symbionts on, and each symbiont produced a different type of antimicrobial peptide or GFP. And then we look at the titer of the bacteria before the gall and after the gall has developed and formed for a period of time. And we were able to identify ones that really reduced the amount of DNA of the pathogen based on this PCR delta CC. So these 2 molecules here, the BP and the G1, were advanced because in our screening strategies, which we can do fairly quickly, we identified them as being able to reduce the titer of the bacteria, the Candidata bacterium Asiatic bacteria in the plant that causes citrus screening disease. Here's an example. Now, this is, I will say, this happens when we get good gall formation and the gall is expressed at high levels. And those are things we're still optimizing. We need to do some better optimization on some of these to get it out commercially. And we've made some good strategy. But when we get a good gall and the gall is expressing, then what we can see is recovery of the plant in terms of reduced symptoms. What we do is we put the gall in the plant. Here's one of our symbiont expressing green fluorescent protein only. Here's one expressing a protein that is active, an antimicrobial peptide that was shown to be active against the pathogen. And you can see leaf regrowth after we formed the pathogen and we defoliate and look at new leaf regrowth, we had a much better reduced symptoms. And if we looked at the PCR analysis detecting the amount of total nucleic acid from the bacteria that causes that, CLAS is the acronym for the bacteria that causes citrus greening. We got a great reduction in detection of nucleic acids. 4 months is the blue and 7 months after inoculation. So we're able to do that. We now are also able to produce symbionts that can express in most of their tissues the desired amount of, or a large amount of our protein that we wanted to produce. And we're trying to optimize that and get it to be at a high enough reproducible level that we can commercially deliver it. And that's what the team is working on now. So we now have them in field trials in cooperator grows under APHIS approval. And we're growing them on trees in the field, both on our research farm and at collaboration. However, I want to take a sidestep because there's one other thing that came out of this while we were doing it. So if you recall, I originally said that we really wanted to be able to form a symbiont on a plant, cut it off, bring it into culture, grow up the cells in mass, and then deliver small amounts back to plants so that they could grow these galls. That has a lot of advantages from a engineering point of view. And so we would bring those cells into culture. And here's an example of a culture on agar media of those cells. And they grow quite well. And these are ones that were producing the M-cherry protein, which is a red fluorescent protein. One thing we noticed is that when we produced it with M-cherry with a signal sequence, they really secreted it all into the media, which I guess you would expect to have a signal sequence that targets the protein to be secreted. But we realized, wow, these are really a great biofactory for producing proteins. So we kind of structured, let me do that. So you can see all that grow in the media. So then we said, well, what if we put these cells in a continuous culture system and we just kept harvesting the media? Here you see M-cherry in the media. Below the cells, we have a continuous culture flushing system that we're looking at here. and all the MSherry is in the media. We can harvest the media, put fresh media in the cell, stay here. We did that for many months, and we can easily rapidly purify a lot of proteins in that work. If we engineer, in this case, we engineered the green fluorescent protein to be expressed in the cells, and so it does not get secreted and it stays in the cell. So depending on what you want to do, we were able to produce a lot of protein. But we can also just do it on the plants. And if we and when these symbionts are producing a lot of protein, or in this case, it's a the beetling product from beets, and they accumulate. They can accumulate to high levels within the symbiont and be isolated directly from the plant. And so from an in-planta perspective, then we said, wow, could you harvest these like you can harvest an apple? And the answer is essentially yes. Here's a plant euonymus that's a good host for the agrostrains we have. We can grow them on the ends of the stems. These are symbiont. These are the gall-like symbionts growing on the ends of the stems. We got rid of all the leaves just so you can see. And they grow quite rapidly. And this is them on grape as well. And if you look at them, depending on the plant type, we get very high levels of GFP protein accumulation in those. GFP is an example of any protein that we could accumulate potentially within these cells. So it could be a harvestable biofactory / biowarehouse. And from my perspective in agriculture, I like this. It might be a way where we could produce biologicals much more cheaply and very scalable in a plant system for use in agriculture. That's always one of the limitations on use of biologicals is the cost. And this may be a much more economical way of producing biomolecules for use in agriculture, especially if we produce them on a plant where if we're going to use them to treat plants or just in a spray, for example, against fungi, perhaps we wouldn't need to do large scale purification, because purification can often be 80% of the cost when people are fermenting things in other systems. And so we're excited about this. It's actually being advanced by a company now that we collaborated with under cooperative research and development. So just to summarize then, our Symbiont technology, I first talked about, we developed it to alter plant phenotype without altering the plant genotype. I mean, you do in the gall, but not the rest of them. And we use that gall as an insulin pump would be used in a person with diabetes, or the plant in this case. Or we could use it as a biofactory / biowarehouse. In the case of altering plant phenotype, we could induce plant defenses against pests and pathogens or abiotic stresses, or alter characteristics to improve nutritional yield, productivity characteristics. This is the vision we have for this. We've been advancing this strategy. We have different ways of doing this potentially, with the idea of developing biodeliverable immunization to plants to emerging abiotic biotic. I mean, that's the big thing that we've been advancing. So I said in the beginning, wow, we'd love to be able to deliver this in 5 years. So initially, we're working with the APHIS, our regulatory agency in the United States, to help us to get. So we understand what their concerns are, and we're conducting the right experiments to help support their getting comfortable with this strategy, so that then the hope is that if we can show them that this is an acceptable strategy where they have great reduction in concern, over the escape of genetic material into the environment or into adjacent crops, because it's just this gall that can't survive or produce things that can survive and deliver the DNA outside of the plant in which it's on, then we could reduce its delivery time. So when a new disease comes in, if we could just adapt it to that disease and that host, and work with regulatory specifically to the genes that we're using, not the delivery platform, it is conceivable we might be able to deliver these in 5 years. So we're excited about that, and that's what we're really pushing for. But there's a number of things that could be done in terms of including crop performance with this, reducing production costs. So, but also there's the idea of this biofactory biowarehouse, where in vitro cell culture, on-plant production, either strategy, and they could each have their own niches where we could be producing, for example, these are just some of the things we're thinking out of the box on: veterinary vaccines, plant protection biomolecules, various industrial biomolecules or biopharmaceuticals and nutraceuticals. So, all right. So, all right. We definitely have improvements yet. We're not fully there, but we're excited about it. And I think we've made good strides since we really started this back in 2018. We have patents that cover the technology now, and we worked with a private company. But there's things we need to do, and I'd like to talk about briefly: increasing longevity of the symbionts, improving export of symbiont-produced proteins of interest, and develop commercializable methods of delivery. Those are hot areas that we're working on strongly. Here's an example of some of the recent things that have been done. This mostly comes out of James Thompson's lab in Albany, California. But one of the things is, if you look at, if we form these galls, these are on citrus. These are with different versions of agro strains. This is EHA 105, that's the strain of agro, and where we introduce just the 4 plant growth regulator genes. They form a nice gall, and you see this at 16 weeks, but if you look at 28 weeks, things start to die on us. Here's another strain, again, with the 4 plant growth regulator genes delivered at 16 weeks, and 28 weeks that dies. However, this is 1416, where we added back some of what are called the BAS genes, and that stands for Transdisciplinary genes, within the wild type tDNA. There's a number of genes that in the wild type DNA of agro that are delivered, and they can influence the quality of the gall form. And when we look at some of those, we're seeing galls that are surviving much longer. This is 28 weeks, and James indicates he has some that are a number of years that just keep, that won't die. They kind of pulse through the growing season, but they stay alive for much longer. And we're excited about that and characterizing those genes now. Another area that really we need to work on, and we've made advances, but we need to make more, and that's on systemic movement. This is a nascent gall, just a new forming gall on tomato. The green you see here is green fluorescent protein, but you can also see the green fluorescent protein moving outside of the gall, especially in the extracellular spaces, because this has a signal sequence attached to it, and it is moving more somewhat systemically. We can detect it in other parts of the plant, but it's much less leveled. We think there's only something like less than 10% moving outside of the gall of the protein that we want to have moved. So there's a number of strategies we've looked at. One of them is I'm sorry, let me advance. And we know a little bit about how to get these things to move. For example, here's GFP when it's delivered by agro in leaves under transient experiments. If we look at under normal conditions, where we introduce 2 different strains of agro, one producing the red fluorescent protein and the green fluorescent protein, and we see if the proteins do not have the signal sequence on, they stay in the cells that they're made. But if we take the green fluorescent protein and add an extracellular targeting signal sequence, all of a sudden, this is the area of infiltration where we added the agro and it engineered the cells. The red still stays in the cell, but now the green fluorescent protein is moving in all the adjacent cells, and we can detect it actually in the vascular tissue of the leaf. So we clearly can get things to export, but does that mean that they go through the process of being moved from the cells that are being produced in a symbiont and eventually move into the system, into the vascular tissue? That's a much more complex idea, and that's as shown here. How do we get it into the vascular tissue? Well, we do know, and this is work that has been done for a number of years in basic understanding gall formation. They're highly vascularized, and this is some of our early galls forming on tomato, where Marco took a picture of the vascular tissue, the white strands moving into just a new growing gall. So we know they're highly vascularized, like this cartoon shows, but how do we move the molecules being made in these cells into there more efficient? Well, we looked at 75 different protein fusions, and where we fused GFP to various exported proteins that are known to move either into the xylem or the phloem. And we just evaluated those, again, using our symbionts as a screening strategy. So here's a cross section of a citrus stem. where you're seeing the blue is the xylem, and just above that or close to the outside of the stem would be the foam area. And here's one where we have a gall producing GFP, and you can kind of see the issue. But some of the fusions we made did a better job of moving things into the vascular tissue than others. And when we advance this to looking at ones, here's a stem, a longitudinal section with a gall growing out of it, looking for ones that move better into the vascular tissue. And we have identified one of all of those that we screened where we actually can detect the green fluorescent protein up in the stem and show with the Western blot of the GFP that we see it up in the scraping of the vascular tissue, most from several centimeters above the gall, and we see it moving. So we're advancing that more now and seeing how that impacts the effectiveness in the field on big trees. Because right now, a lot of the things, well, the evaluations on the big trees in the field is just going on. But we know we need to get better at export if we're going to have robust effects in the field. So that's one thing we're improving. I said we're also improving the commercial delivery. If you look at the picture on the right over here, this is what we currently do now when we create a symbiont on a plant. It's kind of a process that takes at least 15 minutes and people under the tree. And for someone that has, you know, 1,000 acres of trees or 30,000 acres of trees, that's really not going to cut it. It takes too much time. So we're working with companies that have devices, and this is an example of one used to deliver doses, pellets of herbicides to invasive trees to kill them, invasive woody trees. And we're looking to see if we can optimize some of these already rapid delivery strategies for small volumes to see if we can, instead of delivering herbicide, if we can deliver our inoculum for our symbionts. So that's where we're at with that, and we're just in the process of moving that to the field. But I would like to say, in the beginning, when I was on my soapbox talking about how we could develop these transdisciplinary strategies, I realized we needed to also teach the students, the next generation, how to work in multidisciplinary environments and communicate. That's not easy. I know I've done it in the past, and sometimes we don't even speak the same language. And so what we did is we're working with Indian River State College, IRSC, and using their students. They have a fabrication and design facility to make objects using computer-controlled milling machines, 3D printing, et cetera, and designing them. And we're integrating them in teams with biological students, with biotechnology experience, and coming up with a strategy to adapt these devices that are used for delivering different things to be effective at delivering and supporting the development of Symbiont. So we're teaching the next generation how to communicate across disciplines, and I personally am excited. And that's using that delivery device, for example. It delivers these delivery cells that contain the whatever the inoculum is. How do we optimize those? How do we package our inoculum so it's stable and yet can develop in the tree? We've got multiple devices. This is just one example that these student teams are going to start working with. So with that said, to summarize, we have a broad range of uses for this Symbiont technology, and we're focused on treatment against pests. Can we use it to induce natural defenses? That's something we're looking at. Produce natural defense molecules from other plant species that we could engineer to use. We'd like to see, and again, this is part of the reaching out strategy. We know we're working heavily on these bacteria that are flow limited, and work on other bacteria or fungi, insects or nematodes. Those are things we're very interested in seeing if we can identify the genes or the bullets, I would say, for the gun, which is the Symbiont. Okay. But it could also be used to change aspects of plant physiology if the symbionts can produce things that can affect the plant. We haven't gone down that road, but we envision that this could be a very interesting area to evaluate. And then I'd like to close by showing you some very tree galls, I think at least, that I think are really cool. So this is a diversity of insect galls. galls. And I'm asking, can we learn from them? If you saw our galls, they're not so pretty. Well, I kind of like the red ones, but still the structure is pretty gross and amorphous. Can we do something to improve the structure of these galls? And so to answer that, I look at all the different types of galls that different insects can form out of galls. And this is an example of them. And you can tell what the insect is by the type of gall. They each can produce very specific types of galls. It's very unique structures. And how do they do this? Well, none that we've seen so far do it by injecting DNA into the plant. But they do have genes, and in different ways. For example, some of them, the larvae that hatches produces molecules that are secreted and change the plant cells and induce the plant cells to grow into galls. Or the female that injects the eggs may inject something that initiates that process. It's something that right now that scientists are just starting to scratch the surface of. And I think it's fantastic. And as they understand the genes that are involved in doing this and the biomolecules, can we take those and mix it with our galls to effectively improve the structure of our galls? We're starting to work with entomologists that work on galling insects. And the nice thing about our galls is it's so easy to engineer them to produce different things. We can really screen a lot of things. This is something that I think the future of our strategy really this would make our strategy in the future look really interesting and even brighter, and something where I just find fascinating to move forward. So, but again, these are by bringing in entomologists that work on galling insects, bringing it with bioinformaticists, and helping us understand what's going on there and taking those genes and seeing if we can do some interesting things with those in our symbionts. And that's pretty much what I have. I'd be happy to entertain any questions. Marco, can you hear me? Hope I haven't been talking to myself this whole time.
Thank you very much, Robert, for such a nice presentation. And it's really innovative, this approach that you show us today. It would be great if, as I said in the presentation, we can come up with some solutions for our pests and diseases in coffee, because of this issue with chemical control being banned all over the world, all over the places. So thank you, Robert. I will ask a couple of questions first in Spanish, and then I translate it for you in English, if you don't mind. So, voy a preguntarle al doctor Robert, y los que tengan preguntas, por favor, en el canal de YouTube. No, este es Zoom. Entonces, en el canal de YouTube. En el canal de YouTube, por favor, las pueden hacer en español y nosotros aquí las trasladamos, las traducimos. La pregunta que le voy a hacer al doctor Robert es acerca de regulaciones que tiene esta tecnología ya mismo en Estados Unidos o en Europa. Dr. Robert, what kind of regulations you foresee or do these technologies have right now in the US and Europe?
Okay, yeah. So actually, that was part of my reason for going to Europe, was to understand.
I think your audio is off.
I'm sorry?
Oh, no, yeah, I'm sorry. Go ahead.
Can you hear me?
Do these technologies have right now in the US and Europe?
Okay, yeah. So actually, that was part of my reason for going to Europe, was to understand.
I think your audio is off.
I'm sorry?
No, please go. Yeah, I'm sorry. Go ahead.
Are you hearing an echo on there?
Sure.
Okay, yeah. So actually, that was part of my reason for going to Europe, was to understand. Can you hear me now? I don't think anyone is hearing me. Can you hear me now?
I can hear you well, Dr. Robert. Please go ahead.
There's quite a significant delay of maybe almost 10 seconds, but I will try to answer your question. And so, yeah, I went to Europe with the idea of one of the things I wanted to accomplish was to integrate what the scientists understand. And they really embrace this idea, and there's a couple of collaborations moving forward. Clearly, has to be some comfort level with even in the US and in the EU on these technologies. And because it's a new technology, but the advantage is, again, the DNA we deliver does not move into the environment. It can't. So we mitigate all the concerns about the genetic material moving into other into non-engineered plants, for example. There's another point I'd like to make, and that is we can engineer, because we can put close to 70 kilobases of DNA into our symbionts, we can actually engineer pathways for volatiles. And there's a project going on right now to see if we can use symbionts to produce behavior disruption volatiles in pest insects, whether we're attracting them or disrupting their mating behavior, et cetera. And the advantage of that is now we don't have to put them on the crop that is being harvested. We can put them on any plant that could be planted intermittently among the crop, and they could produce volatiles that would affect pest insect behavior. And now we're not even putting these engineered galls on the crop plant. And that's an attractive method as well. I hope that you're getting this. Affect pest insect behavior. And now we're not even putting these engineered galls on the crop. And that's an attractive method as well. I hope that you're getting this.
Okay, we have several questions actually from our audience. So what will be the necessary resources for a research lab to actually implement these technologies in the for the management of diseases in coffee?
So I guess it depends on if you embrace the kind of transdisciplinary approach. I would love, I know money.
Questions actually from our audience. So,
sorry, we're getting constant feedback, so it's been difficult to talk, but I'll try to talk through it. If there's a problem, just cut me off and let me know.
Yeah, go ahead. I think we'll solve the problem right now.
Okay, thank you. All right, so yeah, it depends on if you embrace kind of this an international transdisciplinary approach or what. I know there's some difficulty in funding internationally, but and that was another thing we were trying to address when I was in France, and sometimes we can leverage our collaborations to each get local money. The nice thing about this is the downstream use is fairly low technology. So if you can culture a bacteria, grow it up, and then you just need to be able to take it to the field and inoculate a plant in a very low tech way. So it doesn't require high-end resources at that level. The higher tech kind of strategies in the lab, are identifying genes of interest, cloning them into the tDNA of your Agrobacterium, and that is routine molecular work that anyone can do. We technically now, back in the day, I've been around in this for a long time. Back in the day, we used to say clone by phone. Now we clone by email, basically. We email the sequence to a company, they make the gene, it comes back to us in a couple of weeks, and it's in the vector we want, and we can put it in agro and inject plants. So the resources depend on your level of interactions, but it can be done fairly low tech at the delivery stage.
Oh, brilliant. Thank you. We will worry with you for that. So we have other questions. Any ideas of the type of molecules to use against insect pests?
Yeah, there's a couple. We have a collaborator working on use of very targeted RNAi induction molecules. So short, for example, double-stranded RNA production that's very specific and targets insect pests. We do have some other molecules that, for example, some of the antimicrobial peptides, we didn't realize this, but they have effect on the phloem feeding, the plant vascular feeding insects. We're assuming that, and there's some preliminary evidence, but we don't know what's causing it. We're assuming that there are endosymbiotic bacteria, obligate endosymbiotic bacteria in these kinds of insects, and that the antimicrobial peptides are knocking them out. That's our working hypothesis. We do have little data to support that, but not enough yet. So it looks like there are strategies for the vascular feeding insects like aphids, whiteflies, psyllids, etc., leafhoppers, clodhoppers, that are showing promise and we're evaluating further.
Okay, thank you. I'm going to translate that. Las dos que contestó el doctor Robert anteriormente, una es que para implementar un laboratorio con esta tecnología, por ejemplo, para café, sería realmente baja tecnología. Él considera que no se necesita un laboratorio súper desarrollado, pues una infraestructura muy costosa para esto. Y la segunda, con insectos, él dice que sí hay algunos de los compuestos que ya muestran ser promisorios para insectos y que son de los que están ensayando. Another question is, what other proteins besides fluorescent proteins or antimicrobial peptides have been expressed in the galls? Could you express some kind of interference RNA?
Oh, yeah. And I answered that with the previous answer on controlling insects. So yes, definitely. And then that was to address insect pests. Could they be used to alter plant phenotype? That's something we haven't done yet. But potentially you could knock out host genes to address plant phenotype as well.
Okay, thank you. Let me see other questions. How far is this technology to be used in the control of HLB? I think you answered that on your presentation, but is that correct, right?
Well, yeah, I would say we have the ARS administration is really excited about it, and they've given us a 3-year window to get it ready for commercial delivery.
Okay.
That is the technology. Will it take a little longer for commercial logistics development, et cetera, to get it to the growers? I'm sure that's going to be an issue, but we're shooting to have the technology advance to a commercial albino state. That's our hope.
All right, thanks. And one last question is, do you know any studies about the effects of climate change on the symbiosis? For example, new substances and the degree of liberation to the environment, plastics, antibiotics, herbicides, pesticides. Do you have any? Do you know of any studies about this?
I understood that you were asking about climate change, which I have to be very careful about given the current status of how I answer. But I wasn't clear on how it related to other things. Could you summarize that again, please?
Well, if you think that climate change has had any effect on the symbionts, like you know, if there's any study about climate change affecting symbionts.
Not specifically to my knowledge on our symbionts. I've not done that. But clearly, there are. If you look at Agrobacterium, there are Agrobacterium strains adapted to tremendous diverse climates and just naturally. And so the one thing I would say is in developing a strategy, it's very important to look at genotype by genotype interaction between the genotype of the host and the genotype of agro, and look at how gall formation develops in those. And as we move forward, we're starting to understand the genes that influence that, and we can adapt it. But right now, we just screen agro strains that form the best qualities symbionts. So, for example, some strains won't form a gall at all on the host plant that we want. Well, it'll form galls on other ones, but not that one. Other strains will form very aggressive galls that damage the plant, and we don't want that one. So we pick ones that form with that host, form galls that are beneficial to them, that can be beneficial to the plant. And so if you're concerned about how that environment impacts that, then clearly some agro strains might be adapted to different environments. So as that changes, then you would want to review which strains you're using.
Okay. Two last questions, actually. You show us the possibility of using this technology for modifying flowering patterns. Are you aware of anybody trying to do that right now?
Yeah. Yeah, we are. So, we work on citrus, and one of the problems with citrus or any of these perennial crops is is the juvenility phase when you create a plant, especially in a breeding program, and transgenics, for example, we have some plants that are under permit in our field, and they've been there for 6 years and they haven't flowered yet. And so one of the ideas is if we could produce the right flowering induction genes in symbionts, could we get earlier flowering? Could we trigger earlier flowering in our plants so that we could get for the breeding program, so that we could get fruit quicker and then do the next generation breeding? So we haven't been successful. We've with that using the strategy yet, but it's something that we were interested in, and we're trying as we improve and understand. Now we have a better understanding, and we still need to improve it, but on how to export better the proteins that we make, we will continue to be trying. Other than that, I'm not familiar with anything else.
Okay, las dos últimas preguntas que le hicimos al doctor Robert, relacionadas con el cambio climático. Él dice que no se han hecho demasiados estudios al respecto, pero que ellos están estudiando cepas de Agrobacterium para ver cuáles han tenido cambios probablemente en el tiempo, y que hay que escoger aquellas que realmente sean muy que tengan adaptación y que realmente no tengan grandes cambios ante los posibles cambios climáticos. Y la segunda pregunta tenía que ver con si hay algunos trabajos en floración, dado que pues es uno de los temas, digamos, para café, que sería ideal tratar de regular floración. Y él dice que sí, que ya hay algunos trabajos que se están haciendo en algunas otras plantas, pero en otros grupos al respecto. And the last one, the galls, they seem a bit, how can I say, gross in some ways. So have you tested farmers? What's their impression about like producing galls in their plants, even if they are beneficial, but what's farmers' impressions about the formation of galls?
So yeah, that's a really good question. Of course, I come from a farming family, and the one thing I will tell you, and citrus greening is a prime example of this, and where I said in the beginning, 90% of commercial citrus production has gone away in Florida because of the disease. So any farmer that is producing a high quality crop and making money and buying a new car every couple of years and going on great vacations, the other farmers notice. So the idea is, yes, we do work with the growers. and especially those that are willing to try new things. There are some that say, if this works, that would be great. And we're actually doing it in trials on some growers. And I will tell you, if that grower starts to make money on citrus because of this, that's the driving force. It's not me. They're not going to listen to me. But when they see their neighbor buying new equipment, having a nice new car, they'll try to figure out what he's doing. And that's our strategy is, listen, if we can make one grower make good money, the others will follow.
Great. La última pregunta del doctor Robert fue sobre la impresión que la creación, digamos, de estas especies como de tumores que se creen en las plantas sobre los agricultores, y él dice que ya lo han ensayado y que básicamente es, digamos, tienen que mostrar que la creación, digamos, de esos tumores está a ellos ahorrándoles dinero para el control de una enfermedad, y que de esa forma estos agricultores van a ser el ejemplo para otros vecinos de esta tecnología, ¿no? Que, digamos, no se impresionen por la creación de estos tumores. Dr. Robert, it was a pleasure. Thank you very much for your wonderful talk today. We'll be in touch for next meetings with the team. We really, really appreciate your time and for making this seminar. Thank you very much indeed, and have a good one.
All right. Well, it's my pleasure. Thank you for giving me the opportunity, and I'm looking forward to interacting more as we move forward.
Thank you.
Well, take care.
Muchas gracias a todos los oyentes y bueno, muy buen día para todos. Chao, chao.
Ciao.